Hello, Dr. Shiva. Thanks for joining us in Scouted by Graby. And right off the bat, I'd like to start with this very first question. I mean, as soon as I read about Beez Biotech, I wanted to know how are you guys basically solving this huge problem of infertility that I basically what I was able to get is even IVF is not that successful and ICSI is also not that successful. So how is Beez Biotech solving these issues?
Thanks, Shabaz. Actually, thanks for this opportunity. Actually, from Gravy, very interestingly, that yes, you have pointed out very right question. Actually, if you see that the world of the fertility, if you see the let's say the fertility rate. Country by country in last 20 year is decaying. So if you see the fertility rate is in Europe also, mostly in developed countries, in US, in Europe also. Now it's started in India as well. If you see China and Japan, they are suffering with it as well. So how the assisted reproduction that we call that involves IVF, ICSI processes and IUI processes as well. So we call it assisted reproductions, medically assisted reproduction. So these processes comprises these three techniques where the patients, they are suffering from the infertility, they go and seek this kind of medication from the practitioner, where they execute these methods to help them. But where is the problem? The problem is, if you see that the average success rate of the these methods, including, okay, I'm not going to involve the IUI at all, because for me that is shooting in dark. I personally not recommend anyone to go for the IUI. So that is, but if you're going for even for the IVF and ICSI processes, that is kind of the success rate you see is averagely over the, it's around 22% the worldwide. And that is not sufficient if we going to counter the infertility, because if you see, the WHO already have kind of provided us kind of alarm that, okay, it's 1 in 6, they are already in worldwide, the people already were infertile. So it's increasing, and if we have this kind of the treatment that we have, it's have this kind of mediocre success rate, that is just around 20-22%, that is not sufficient to counter this global challenge. So here we are now. How Beez Biotech is trying to fill this gap, actually. So there are various aspects, there are various reasons that why we have such a low success rate, and one of them is the way that we select the reproductive cells in laboratory. And that is the one reason, one major reason actually. And there's other reason as well, that is, it's also related to the female ovarian response, because the host, how the female hosts the injected embryos or sperm cells, it matters also. So it's also the how the ovarian response is there in female. Also there's other aspect, genetic aspect is also there. And also there's a problem with the lab standardizations. Lab standardizations means that so many laboratories, they not follow the same protocol, means that very standardized protocol, and where the human error happen in embryo transplantations or in cell selections or even ICSI processes. So these also methods, these also the problem over there. But when it's come to the gamete selection, reproductive cell selections, if I say, the problem is over there is little bit big, because when it's come to the oocyte maturations can be easily seen from the microscope, can be easily, let's say, also there's how the rigidity is there, softness is there, it's also matters, so it can be assessed, let's say, properly. But when it's come to the male reproductive cells, it's very, let's say, the motility is there, it's, they are motile, they're moving with the quite good speed, and also they have the let's say the DNA information is remains in a tiny 8 micron head. And that is something is using just microscope is we cannot diagnose it properly. So how Beez Biotech is trying to facilitate this outstanding solution that we have the solution that is exactly replicating the reproductive, natural reproductive environment that is happening in nature. So what is the process exactly? We call it biomimetics. So exactly what is the same process is happening in natural fertilization process. We replicating the same thing with our technology, rather than having the cell selections based on currently the centrifugation based method and all that.
So what I'm able to understand is the key is in the right way of selecting the right cells to increase the success rate of MARS, basically medically assisted
Actually, as I
reproduction, right?
mentioned it out, yes, thanks. Actually, as I mentioned it out, that it's not. There is a various aspect. There is how the female ovarian response is also very important. Also very important. The lab standardizations is also very important. So it's genetic aspect. Okay, there is a challenging because there's some infertility is genetically, let's say, indulge as well. So it's let's say in these things that we are, apart from the sperm selections, I would say, apart from the gamete selections, I would say. But these three things, we are also, it's not only we are working one aspect in a company, but our first technology that we have developed, it's related to basically the sperm selections and the quality sperm selection. So what is the problem? And that also little bit mitigate the error associated with the let's say lab standardization. So for example, some laboratories, they have different diagnostics methods. Some laboratories, they following, so I'm going to explain you that how they select the sperm cells in a laboratory. What is the process? I think that will be I think
It will be interesting, yes.
that will be, I think, reasonable here to highlight. So what is happening if you are in, let's say, if the patients, when they go to the clinics, what they're doing, they go for the diagnostics for the male. Actually, I'm talking about male only. So they go for the semen diagnostic first. So there is a various method that is available. Some labs, they simply, the standard protocol they follow, it's the motility and also the cell count. So what is the cell count they have? What is the motility they have? What is the motility? Means that number of moving cells in a semen
Yeah.
samples, basically. And also the what is the, how many, let's say, the pH, what is the pH of the semen sample, and what is the number of dead cells, I mean the percent of dead cells, the percent of the motile cells, percent of
Yeah. How
progressive.
many of them are swimming and
Exactly.
how many of them sleeping? Yeah.
Exactly. So exactly, in the layman term. So in those things are there, they assess with the microscope. And okay, there is another automated systems that is, it just, you take the semen sample and put in a, let's say, in a glass slides, and just like a cassette, you put in a system that give you the automated analysis where the trajectory, the dynamic of the sperm cells, they give the dynamical parameters, the velocities, and how the sperm cells are
Mhm.
beating and all that. So those parameters are also there. But look at the picture that over there, it just, the motility is not only the criteria for the assess the fertilization capability of the sperm cells, because that is not only the marker to when the natural fertilization process is happening. uh, Apart from motility, there is a lot of microenvironment there that plays the role in a natural fertilization process. So okay, this is the one thing. That's different laboratories, they're following the different protocols. Some following the CASA, that we call computer assisted semen analysis, automated analysis means, and some following the manual processes. And for the sperm selections, what is something that really drastically I do not like it at all, is like called centrifugation based process, where they take the semen sample and they centrifugate it, just like old time with the blood samples and all that. Blood sample to, they centrifugate it to separate the plasma, white blood cells, red blood cells, and platelets and all that. They would do the same process, not exactly same protocol the is optimized for the sperm cells, for the semen sample, but they do the centrifugation to separate the plasma and the sperm cells, and the quality sperm cells. So in that those processes is not normal for the semen sample, for the sperm cells, let's say. And in that way, somewhere, okay, there's a semen raw mother, some mother sample already have some DNA fragmentations, but also it can cause to the damage to the cells, the centrifugations, because it's not normal process for sperm cells.
Mhm.
And that DNA fragmentations leads to the fragmented cells. Actually, research has shown that the fragmented cells actually can fertilize the egg, but it can develop into the, let's say, the gene expressions alterations can be there, and it can lead to the miscarriages when the embryo implantations happen with this kind of samples. So to mitigate this error, what the technology, as I told you, we have developed, we are mimicking the same natural fertilization process, because sperm cells, when enter to the female reproductive tract, it have certain mechanism to pass certain levels, let's say going from cervix to uterus. It have a certain physical cues which actually govern the migrations of the sperm cells from, let's say, from vagina to the fallopian tube. And those marker actually we incorporate in our technology to select high quality sperm cells.
This was a very elaborate explanation, and thanks for going taking me through this. So, what is the underlying technology that is helping you improve this process and basically maybe replicate this natural process? Is there any underlying technology that your Beez Biotech has researched on their own and they've come up with this method? So, can you tell me a little bit in detail about that as well?
Okay, you mean that I need to go to the technology detailing? You want that?
Yes, yes, yes.
Okay.
That is, yeah.
Okay, so there is a various aspect. I think, I don't know, you are aware of the certain kind of keywords in what is your background, if I ask you?
Oh, my background is marketing, but I'm aware of some of the terms because when I was researching, I had to read some of the stuff. But feel free to use any industry terms because this interview, whenever it goes live, it will reach the right audience,
Okay.
people who really understand these terms.
So if you see that there is a if I explain you in a little bit layman terms, the
Yeah,
how
I'll
the
appreciate that.
natural, yeah, how the natural fertilizations functions. So natural fertilization function in a way that when the let's say sperm cells enter to the female reproductive tract, it has to pass through the female vagina, and which is very acidic environment for the sperm cells actually.
Yes,
and
it makes
and
things difficult for the sperms to
Exactly,
travel and all. Yeah,
exactly. And in that time, let's say if you see that the during the intercourse process, there is a some biological fluid secretions is from there. So the sperm cells, they have to be they have to leave that acidic environment very fast, because otherwise they will die. And they have to leave that environment very fast, and also they have to move against the flow, because there is
yeah,
some biological fluid secretions is happening. And those secretions, it's actually in that process there's a lot of bacteria and viruses. It get automatically filtered. Actually is those conditions to protect the to the females from the other
yeah,
let's say those external
yeah.
say
Any
biological, exactly,
infections or anything
exactly,
like that.
yeah,
Yeah.
that we have. So in that way, the sperm cells, they have to move counterflow to enter, to pass through the cervix region and to go to the uterus. And once they pass through those once they go to the uterus, apart from there will be other attack, because immune systems also attack and trap some of the cells. But apart from that, there is some additional marker, because if you see during the ovulation time in a female, the body temperature, not the body temperature, the near to the fertilization side, the temperature is a little bit higher, and that creates the kind of the gradient. And sperm cells, they have this very specific receptors. They recognize these change in temperature, and they migrate towards the oocytes. And also the oocytes, they release very specific biochemical to attract the sperm cells and invite to them for the fertilizations. So actually, if you see, these are the step by step the several biological mechanisms that playing role to favor the natural fertilization process. So what we simply trying to do, we have developed the box that creating the temperature environment and the chemical environment to provide the environment that is actually for the favoring to the natural fertilization process.
You're basically replicating
Exactly.
the internal system in virtually in a box.
Exactly.
Am I
And
getting it right?
exactly. And there is one disposable chip that we couple with the box. And the disposable chip is based on microfluidics that provide you all these flow conditions, the shear stress conditions, the flow conditions, that is actually separating the filtering, let's say, the non-sperm cells or bacteria and viruses, and allowing only those sperm cells which can move against the flow and reach to the other side. So it's like a to separating those cells. It's like, okay, if you passing this flow condition and reaching from point A to B, you are selected. You are, it's like that.
This very visual representation of things. Thank you for that. So now I'll move on to on the business side of things a
Yeah.
little bit, because now I understand the technology very well. So who is your target audience? Basically, I read, and basically I think from the website what I'm able to figure out is you are enabling reproductive laboratories by providing them your technology so that they can give better results to their patients.
Yeah.
So is am I have I got that right, or is there any other
Yeah, I think
expansion strategy you are involved
yeah.
using?
So IVF laboratories, the all these let's say there is some now the IUI laboratories also opening from the midwives and all that. So they are actually, the IVF laboratories are either they're working in a big scales or small scales. They are our primary end user, and it can be extended to the sperm bank, and also the technology in the future can be used for the veterinary purpose as well, like animal breeding and all that. But so far we have developed a project that is very human centric, and we want to really solve the this problem. And that's why we all the projects that in Beez Biotech that we are addressing is basically, that is, we want to solve the frustration associated with the only with the human reproduction, and we don't want to diverge from the as I pointed out in a first four reasons, let's say the ovarian response, the lab standardizations, and also the, let's say, genetic problem to the infertility, and also the selections of the gametes. These are actually four pillars, and we are developing the projects around that. And all the clinics, they are working in this, trying to practicing the medical assisted reproductions. Our product and our services is for them.
So where are you on this journey? Basically, the do you have is your proof of concept working, or do you have already started selling it to laboratories, or have you seen success rates already? So where are you in this stage?
So if you see that the kind of normally a biotech or pharma technologies take 5 to 7 years to go to the market,
Yeah.
and we are
Mhm.
just now, we have a 3 year old, we are 3 year old, if you see. So in that way, that I think we had a very good speed. So our technology is now is already, I think you aware of the one term called TRL, technology readiness level. So
Yes, yes.
that we need to, we are simply, when we're doing some R&D activity, we implement this kind of let's say scale to assess the maturity of the technology. And we already developed the proof of concept. We already even we have delivered to minimum viable product. That is,
Mhm.
it's not industrial product, but it is the product that where our end user can operate. And we already have tested these minimum viable product in 3 laboratories in France. We are not selling it, but it have provided us the kind of real time testing of the technology that we have developed. And I can tell you that we are getting very good results on that. So
Mhm.
in terms of the technology readiness level, I call it TRL 5 to 6, where technology is almost knocking the door of the commercializations. So setting up the, from here, now we need to get the enough funding to get the regulatory approval done. Regulatory approval,
Mhm.
it comprises the CE marking for the this medical device that we have developed, and also the FDA approval for the US commercializations. So we are now seeking the fund to move ahead with our industrial productions and commercializations of the device, comprising the
Yes.
regulatory approvals. Yeah.
Thank you for already mentioning it, because that is what I was about to come to. Like, where are you on the regulatory scale? Because I understand medical technologies, they take even longer in sometimes in regulatory approvals, and this the process is very complex. Specifically, the FDA approval process.
Yeah.
So, but again, good luck with that, and I hope you guys uh, go through this successfully. So what challenge do you see? I mean, do you see any challenge that, okay, maybe this is a hurdle that we'll have to tackle before we go into commercialization, or maybe once you go to commercialization, then what's on the horizon, that do you see that this is going to be the next challenge that we have to solve?
Challenge means in terms of the technology or in terms of the enterprise?
I'll appreciate both, maybe both in technology terms and business terms as well.
Okay, so the technical terms are what I will talk about. That normally what we have delivered to the clinics. I think one thing that now we are seeking the let's say what we have delivered. They really like it. They really like the concept. They really like the way that we are addressing this issue, because this is obvious process. It was existed since the beginning. I would say natural fertilization process is not something that is mysterious. It's a very well studied phenomena biologically, but replicating, yes, replicating that phenomena, it's challenging. And what my ideology is this: for develop something, some technology in a way that nature solves all these mysterious problems very nonchalantly, I would say.
Hmm.
Effortlessly they solve these problems, and I simply try to study it and replicate it if I can. In a way, yes, sometimes it's very challenging to, because nature, the way that it solved the something, it's very challenging sometimes. And yes, so we have done it and we have delivered it, and it's liked by the clinics that we are testing with. So in terms of the technology, now we are really trying to secure the fund to produce the industrial batches, because that will provide the real testing with our competitors, with our the technology that they are already using. And that is the something we are trying to address in terms of the technical point of view. Coming to the let's say sometimes in a business aspect, yes, I think it's happened with the Beez Biotech nicely, that okay, we really address the commercial value of the technology very well. But sometimes when you are trying to, it can be happen to any company, sometimes when you're trying to develop something, and you really develop something, and then you go to your clients and they say, okay, I need these modifications and I need these modifications and I need these modifications, and those modifications can cost you the big amount. So I think for the startup it should be like that, that really taking the considerations step by step to validating their technology in the eye of the customer. Because that is something that we have, I think I can say you that we have done since the beginning. So that's why that the point that we have reached, I think that is the technology is liked by them, and now we are seeking the fund and to enhance the team to achieve our let's say commercials and let's say the business goals. So far we have received some funding from the European Commission to develop other projects, let's say, but those are the projects that we are visioning to develop other products in line with the product EVA that we have developed for the sperm selection. So coming to your back to your answer, that what you are trying to seeking, that what is the challenging, yes, the challenging is getting the, that's a money out of the someone pocket. It's really challenging when we are, even still
Definitely.
you have, maybe you have the very technology is very good, you have very nice feedback, very but sometimes it can be challenging to get the fund, especially the private fund, when it's coming to the normally the regulatory for medical devices comprises very high cost. The production comprises very high cost. So the plan to commercialize one devices for category 2A or 2B, it's like 1 to 2 million can be over there just for the European market. And getting this kind of big fund for the startup like us, yeah, it's challenging, where you have the small team and you
Hmm.
have the not a lot of resources, I would say.
Okay, that's interesting. Dr. Shiva, have you seen any success from public grants and government grants, acquiring funds from there,
Yep,
or has Beez
actually,
Biotech tried?
if you see that, I actually am the recipient of the Marie Curie Individual Fellowship, and Beez Biotech is impactful outcome of that fellowship. So it's a grant for 200K that I wrote and received, and I joined Europe. My ex, let's say, employer called Cherry Biotech as R&D manager to start this project. And after that, also this year we have secured another that is also for 200K, but developing different project, because as I told you that we are not only one idea and one thing that we are working on. We are
Hmm.
scientists. I'm scientist by the professionally, let's say, I would say. And okay,
Yeah.
I become entrepreneur, but the thing is, I have, as I told you, that we want to solve the, to provide the, there's not only one aspect that constraining the success rate of the assisted reproductions. We are trying to see the each and every pillar, and then we are trying to how we can break these pillars one by one to provide you the solutions, either service or the technologies, so that we are working on. So the one project that we win this year, I think it's belonging to the one of the studying the fundamental aspect of the how the sperm cells interact with the oocytes. So that we are studying this through this project. So we have secured almost like 520, including public and private, let's say, funding. So we have, since the creation, since the inception of the Beez Biotech, we have secured almost approximately 520k euros for the Beez Biotech. Yes.
That's good to know, and I wish you good luck again
Thanks.
for your points as well. So I had this question now that you mentioned that your proof of concept has also been liked by a lot of your target audiences and other people as well. So how easy it is for a laboratory or for a clinic to incorporate your technology into their existing processes?
Actually, it's just I'm replacing the sperm selections currently for particularly speaking to the in terms of the EVA. So I'm
Hmm.
not going to replace anything from their laboratories. I'm just replacing the currently what they have. They have system, the centrifugation based systems, and they have the technicians that who are performing the sperm separation processes for them. So simply I'm replacing that system from their lab, and rest going to be as it is. So it just I'm replacing the sperm selection process with the one technology that we have developed. In the future, yes, we are working some other technologies as well to provide in the services, to provide in the better diagnostics. So those, when that is not even the proof of concept stage, but those, it's they are in a process, but when they will be developed, it will be there. But so far with the one technology that we have developed, it's just replacing the one simple step from their laboratory, how you're going to select the sperm cells from the semen sample, and that's it. That process will be different from the what you are doing at the moment in your laboratory. That's it. And rest as it is, you're going to be they're going to execute as it is, as they are.
And as I've seen, because I've talked to a lot of founders who have worked in like on a completely new solution in a different industry, and it sometimes it ends up being that you're not just providing a solution, but you also need to train your audiences
Yeah.
to use your solution. So is that the case with Beez Biotech as well? So basically, to incorporate your sperm selection technologies, you also
Yeah.
provide some training to them.
Yeah, actually, indeed, yes. I think that with the Bees Biotech, what we have developed, it will be the protocol is very simple. What we are simply wanted to do, because the centrifugation based protocol is very simple as well, and it is quite fast for them as well. So it's once I think the what we have learned through this entrepreneur venture, that what you need to provide the better value propositions to them. So, for example, that I'm not going to increase the protocol complexities. I'm not going to increase the protocol timeline. So with that timeline, without having those complexities, because it's not a multi with the centrifugation, you need to do the multiple time. Let's say centrifugations, and it's multi manipulation processes. So I'm not going to just going to tell you that okay, insert one side and collect from the other side, and that's it. So that is a very simple process for them. I hope that with the pipette they can just put the semen sample
Hmm.
and collect the simple semen sample from the other side. And these things it can be written in some kind of, let's say, very small booklet in a protocol with the product packaging itself. So it will be not very let's say, they are technicians, they are very educated. My end user, they are very educated people, so
Hmm.
it will be not hard for them to adapt this kind of changes.
Okay. And of course, Dr. Shiva, as you've already mentioned that you are by nature, by your work and by your background, you are a scientist, and this uh, once you started Bees Biotics, and that's when you entered into entrepreneurship space. So, and a lot of work that am able to see of Bees Biotech is dependent on all the research work and all the preparation that you and your team might have done. You have lots of academic background as well. So how are you protecting your ownership? Like, what are your views about IP in this case?
Yeah, the first case that we already filed the French patent. We already, let's say, filed the PCT applications already for the technology that we have developed. Now we are willing to extend the PCT application to the EU, US, and China. So that's the first step is that to so far, as you said, that that I don't find a lot of informations on a website. I don't find a lot of informations
Mhm.
on there. So yes, that was the one reason, because the website is a little bit old now. We need to upgrade a little bit, because our French patent is already public, and then PCT applications is already public as well. So now we can make it, and we are writing now manuscript to publish our data as well to the scientific journal. So now we were going to upgrade a little bit more to provide more insight about the technologies and all that. But as you said, that we were kind of under the radar. We are trying to be under the radar until our patent applications was not in the
Mhm.
public domain. So, or the other way also, if you're going to talk to the investors or talk to some people where you're going to explain about technology in a very deeply way, because sometimes you need to explain your scientific findings when you are doing the testings, like, okay, what is the efficacy of technology, how the shear stress is working, what is the fluid profile in the microfluidic channels, and all those things. So how it's happening, if you put in the height difference is this and a height difference is that, for the flow, for the pressure flow. So all those informations, let's say, sometimes it from the investor committee or from the, let's say, some scientific community is scrutinized, let's say, very deeply. So there we sign the NDA, non-disclosure agreement. But so far, now it's we already filed the patent, and the patent application is already public. So now the you will see more information on website in
Yeah,
upcoming.
yeah. And actually, information on website, it's again for, as you mentioned, you're under the radar, and of course, a lot of startup who are trying to solve such a critical problem and coming up with a new solution. It's common. Marketing is not on their priority list initially, and solving the problem is on the priority, and that's why this information is not available. It's not just about the website; it's also about on other sources,
Yeah.
and that's the whole goal of our Scouted by Gravy, to bring out these innovation out there, and basically spreading the word about your innovations, and specifically about startups who are under the radar at the moment. Okay, so with this, I'm nearing to the end of this interview. And so far, I have so much fun talking to, and I've got to learn a lot of stuff. So, Dr. Shiva, like during this, again, I'll jump back to the previous line that you're coming from a very deep academic background where you have like experience in microfluidics and bioengineering, and then you decided to start your research in infertility, and now you are like way beyond. Where beyond just an idea, you have an MVP ready, and you're also running a company. So during this all this journey, did you ever feel that maybe this is not going to work, or maybe this is going to be too difficult to scale, or again those that difficult moments? Have you gone through this? And of course, I'm not asking like have you gone through, because almost all the entrepreneurs go through them. So how do you basically take care of such things? Do you ever feel like that?
Actually, if you ask me about the technical point of view, that the technologically belief that I had, it was very strong. You can say that, okay, either it's I'm crazy, or I'm very brave. I don't know. You can say on that, but I never doubt on a concept. I never doubt on a hypothesis that I have, that okay, I'm going to replicate that this step to select the. Yes, it started delivering, and yes, it was the some steps to achieve the, let's say, the customer desiredness. Because when we I told you already, when we start testing the technology with our end user, they have provided, okay, in the beginning we were not receiving the cells enough to that they can execute the even the processes, IUI or
Hmm.
IVF or XC processes. So we have done some modifications in a chip. We introduced something new in the chips to let's say to achieve the, let's say, the commercial goals, I would say. But at the concept at its wall, because nature is already selecting. So I would firmly believe
Hmm.
on that. Okay, if nature is doing like that, I'm, let's say, follow this process as it is. Yes, I'm going to, because in the nature also there is only one cells fertilize the egg, and there is a
Hmm.
very less percentage of cells they reach to the fallopian tube. And this is a there is a one kind of myth in a clinical setting, that okay, the concentration matters. But no, it's the ultimately concentration does not matter a lot. It's only the qualitative cells, the quality of the cells. So we have gone through this process. But I believe me, I'm that kind of guy. I had never doubt on a technical side, and I had never doubt on my technical skills that I'm not going to solve it. that was there. Yes, indeed, on an entrepreneurial side, on a business side, I even still keep testing myself. I keep testing myself. I doubt on my skills every day on a business side, and that's give me the little bit boost to learn, also to keep motivating, to keep finding the new things about the businesses. I'm not a natural business person, okay, but I'm trying to learn how to do the business
Run a
books,
company
how
and
to exactly
navigate all this.
exactly how to project your
Mm.
company in a let's say how to find the fundings, how to activate the channels for the your disseminations of your results, how disseminations of your finding, how to approach to the people on working on your soft skills a little bit to so these are the point that I still doubt, and I it's my job also to keep doubting on me as well too, because I want to improve step by step. So those things I'm keep doing it. I'm not saying I'm not doing it. I keep doing it. But sometimes you know you cannot do everything yourself, like say working on a business, working on a technical side as well. So of course it's required the I have just limited working hours as well, but yes, it requires certain times, but I'm really enjoying it. As I told you, on a hypothesis, on a concept, I had never had a doubt. Yes, building a biotech venture, because challenging is that the building a biotech venture from the scratch is something challenging, I would say. It's not very when you achieve something, the people also come to you, okay, I will buy your technology, or I will have, because I don't know, I have not seen so many startup, that biotech startup or medtech startup starting from zero and making
Mm.
it big. So I have not seen it, because there are so many challenges in terms of the regulatory, in terms of the getting the all the licenses approval, maintaining the patents and all those things. It's very expensive, and maintaining the team as well. It's quite expensive also
Mm.
for a startup. So those challenges, yeah, we are into it and we are up to it, and let's see how we going to solve it.
Yeah, that's a very positive way of thinking. And of course, this is a common skill I've seen in a lot of entrepreneurs and a lot of startup founders is very less of them have doubt about the success. So the confidence is what keeps them driving towards and solving these challenges. And I believe I can see that in you as well. Like you'll keep on solving these challenges, and of course it's a journey. But thanks for sharing it with me and our audience as well. And with that, Dr. Shiva, this was my last
No,
question. I'd like to.
just adding on this point as well a little bit. I think it's to me, I'm doing entrepreneurship, and let's say the it's not being a successful entrepreneur or unsuccessful entrepreneur. To me, the success rate is very low when it's coming to the success. How you define entrepreneur? Let's say success rate is very low for the, because in mostly only, let's say averagely only 3 or 4 company they survive. And let's say in long run, if we talk about 5 to 7 year of entrepreneurship, only in a out of 100, 97, 98% let's say company they fail. So it's not about success or unsuccessful entrepreneurship. Of course, there are some. I'm not saying that entrepreneur they cannot be bad or they cannot be good. It's the way that they can be good or bad as well. But to me, the enjoyment of the process and so much, even failure will give you the so much learning. Because believe me, in last 3 years, what I have learned, I would not have learned in my academic career for sure. It's like so many aspects, the humanitarian aspects, also the technical sides, knowing to my end user on productions, regulatory, and everything. Maybe if I'm continuing my academic research, I would not have those information that currently I'm a let's say I'm comprising. So this is the journey that it's really wonderful, I would say, and I'm enjoying this journey so far. And we'll see
Mhm.
that we'll successful or not successful, only time will tell. But I think there's something that I want to deliver. I will be really proud as an entrepreneur. As a scientist, I'm really proud if I'm from the bench, I'm delivering something to the market. And as an entrepreneur, I will be more proud if is this product that I have delivered to the market is creating the some social impact and also some capital impact to the place that I'm with. So that's the kind of thing that I'm looking forward to.
That's a, again, thanks for sharing this, and I wish you good luck and your team to your team as well. I hope. Actually, I can see already that you guys are solving a very critical challenge, and you're on the right track, and you have the product developed, and you're very clear about the market and the way you are trying to portray your product.